Hello, folks. This is the speaker of group number 10, and I'll make a presentation about UV vision spectroscopy. First, I'll talk about properties of UV vision spectroscopy. As you can see, on the left is picture of UV vision spectrometer, and on the right is schematic image that shows how spectrometer works. Briefly speaking, UV vision spectroscopy uses light attenuation that occurs when light passes through and interact with analyte. The light comes out from the lamp and is concentrated by the lens. Then it passes through a monochromator consisting of prism and slit. Since the light consists of various wavelengths, only certain wavelengths is extracted by monochromator, and the light interact with sample. After all, instrument shows result of interaction. So what is the reason we use UV vision spectroscopy? UV vision spectroscopy is usually used to measure concentration of analytes. As shown in UV vision spectra, using a difference in observance of solutions, we can figure out difference in concentration of each solution. There are several advantages of UV vision spectroscopy that it is cheap and easy to use and consistent accuracy despite the small size of the sample. However, it also has disadvantages, including it results in broad spectra, and the analyzed solution should be diluted to apply Beer's law. Now let's talk about how to calculate the concentration of analytes and what happens when the light is irradiated. Beer's law tells us relationship between observance and concentration. When the light is irradiated on the sample, some is reflected or transmitted. In this process, a power of the transmitted light is measured, and then the ratio of initially irradiated light and transmitted light is calculated to get a concentration information. But you need to remind that this equation is only applied for a diluted sample. This is because if the sample concentration is high, random interactions between the analytes may occur when light is irradiated on the sample, resulting in a nonlinear relationship between concentration and observance. The observing process is conducted by 4 different electron transitions. When the light is shed on the sample, according to the energy level difference, a light which has specific range of frequency is observed to excite electrons from LUMO to HOMO. Therefore, the amount of observed light tells the concentration of analyte, and the wavelengths of the observed light tells us chemical information of the analyte. As I mentioned, the disadvantage of UV vision spectroscopy is a broad spectra, just like this. This is because UV vision spectroscopy is preceded by excitation of electron from LUMO to HOMO. In this context, there are many vibrational energy levels in both LUMO and HOMO. It means that electrons can exist in many different energy levels, resulting in many kinds of absorptions of wavelengths. This character of bonding levels results in broad spectra of UV vision spectroscopy. In this chapter, I'll talk about components and operation principles in detail. Now I'll tell you about the structure and operation principle of the spectrometer. The spectrometer is largely composed of 3 components: a light source, monochromator, and a detector. The light source has a different spectral irradiance depending on wavelengths. For the deuterium arc lamp, it has a good light intensity in the UV area, and the tungsten halogen lamp has good light intensity in the visible area. So usually 2 are used together and used as a dual lamp system. Therefore, sufficient amount of light can be obtained in all areas of UV and visible wavelengths. In the case of an old or poorly performing light source, noise may be severe, and when the light of the deuterium arc lamp is over, only the spectrum of the UV region may generate noise. And monochromator is a device that extracts only monochromatic light by placing slit in front of and behind the dispersive device that disperses the light source by wavelengths. The easiest monochromator to think of is a prism. The prism achieves dispersion due to the difference in the material refractive index according to the wavelengths. At this time, the degree of refraction is determined by the material and the temperature of the prism. However, in real device, diffraction gratings are often used. In the prism, refraction is used, but in diffraction grating, constructive and destructive interference occur due to diffraction. The diffraction grating uses the difference in diffraction direction for each wavelength due to interference. The degree of dispersion is not determined by the material and temperature, but by the spacing of the groups. That is, the group density is important. Prism has a problem that it is affected by temperature for using extra equipment, but diffraction gratings are widely used because of the effect of temperature is small and the degree of dispersion can be controlled by an easily adjustable factor in the actual manufacturing process called group spacing. It is an important component related to the resolution of the spectrum. The last element is the detector. The basic principle is that when light is irradiated into a glass tube with photocathode and anode installed, electrons are ejected and an electrical signal is generated and detected. When light is when the light is irradiated to the photocathode, which has a small work function and easily emit electrons, electrons pop out, and this is called primary electron. Just like this. Primary electrons pass between electrodes called diodes with voltage of 100 to 300 volt. The electrons accelerated as they pass through them. The accelerated electron hits the diodes more strongly, and more electrons are emitted. Many of the emitted electrons are accelerated again, hitting the diode more, and the electrons are released. And this process is repeated. This series of processes is called secondary emission, and electrons emitted through secondary emissions are called secondary electrons. And this process is repeated between 10 to 20 diodes, with the first one primary electron producing about 1 million secondary electrons later. Since electrons are emitted from the material by light, they are called photoemissive, and they are classified as eternal photoelectric devices. Next is the charge-coupled device. In CCD, electrons do not release out of a material, but when light is incident, electrons and hole pairs are created, inducing current and detecting as an electrical signals. This is called an internal photoelectric device. By applying a voltage on the oxide layer, electrons can be collected over a certain period of time. Instead of detecting electrons generated in real time, you can read the electrons generated longer and few seconds at a time to see the signal larger. Also, since it is based on the semiconductor, it can be manufactured small and integrated to be used as a multi-channel detector. Next, we will briefly introduce the equipment equipped in the department, the solid spec 3700. This equipment additionally uses an argon fluoride excimer laser that can detect deep ultraviolet. Also, the type of monochromator is diffraction grating mode. The important thing is the detector, and the equipment basically uses a PMT type detector. The solid spec 3700 DBUV have 3 detectors, which cover the range from ultraviolet to near infrared. The sensitivity in the near infrared region is significantly enhanced by using both indium gallium arsenide and cooled lead sulfide detectors. Highly accurate and highly sensitive spectra are obtainable from ultraviolet to near infrared. Conventional spectrophotometers have used a PMT photomultiplier tube for the ultraviolet and visible region, and a lead sulfide detector for the near infrared region. However, neither detector has much sensitivity near the wavelengths of 900 nanometer, preventing high sensitivity measurement in this range. The solid spec 3700 make it possible to take high sensitivity measurement in the switchover range by using an indium gallium arsenide detector. The advantage of the equipment is that it has less noise compared to the model with two detectors, as mentioned earlier. Another advantage is that large samples can be measured non-destructively. The three-dimensional optical path enables non-destructive measurement of large samples without having to cut them smaller. In the optical paths of previous models, light only traveled horizontally, but these models include a three-dimensional optical path with light also traveling in the vertical direction. Samples can be placed horizontally, which makes it easier to place large samples. In this chapter, I'll talk about applications. Speaking of its application, the main strength of UV vision spectroscopy is the versatility with simplicity. Frankly speaking, unlike other characterization methods that we have dealt with, UV vision itself cannot give neither specific nor unique information about the sample. However, due to its simple measurement procedure, UV vision spectra can be regarded as quick but versatile supporting information. For the aspect of qualitative analysis, as you all know, chemical species that the sample contains can be distinguished. As the top tables show, spectral data for tons of chemical species are already well known, and these data are easily accessible. Simply by comparing the peak locations with this reference data, assumption for chemical species is quite possible. Moreover, not only for complete molecules, if the sample molecules contains unique functional groups called chromophore, their distinctive absorption peaks also appear, just like this. Using these peaks, assumption for consisting functional groups is also possible. Moreover, for not only chemical data, but structural data can also be obtained. This figure shows spectral data for different gold nanorod samples conjugated with nucleic acids. Due to surface plasma resonance, UV vision spectra of gold nanostructures can be affected by their dimensions, such as aspect ratio. In addition, if there are any conjugated molecules, peak shift also occurs. Based on this information, UV vision can be used for obtaining and cross-checking structural data, especially for organic and inorganic hybrid materials for which applicable characterization tools are quite limited. To tell the truth, for not only qualitative analysis, but also quantitative analysis is still possible by using UV vision. As already mentioned, by applying Beer-Lambert's law, concentration of the analyzed solution can be measured. For a diluted solution, observance increase in proportion to the concentration of analyzed solution. Since the length of the cuvette can be easily measured and absorption coefficient for analytes are also well known, estimated sample concentration can be easily obtained by simple calculation. For non-dilute solution, Beer's law cannot be applicable due to interaction between analyte molecules. In these cases, drawing calibration curves using curve fitting algorithm might be helpful. Nonetheless, in both cases, estimating concentration of samples can be done by the simple manner. Since this versatility, there are some enhanced characterization method based on UV vision spectroscopy. One of them is circular dichroism spectroscopy, or CD in short. Before to deal with CD itself, it would be better to deal with circularly polarized light beforehand. As shown in the top figure, circularly polarized light, or CPL, propagates while its electric field vectors are rotating. Therefore, when CPL is exposed to the sample, the rotating electric field interacts with the sample's 3D structure. More specifically, since there are two kinds of handedness, left-handed and right-handed, if the sample has 3D structures with specific handedness, such as helical structure, then the sample reacts differently whether the CPL is left-handed or right-handed. As shown in the right bottom figure, Cherer samples energy levels get split when CPL is irradiated, and by measuring this energy split, the sample's enantiomeric properties can be measured. This can be done easily based on UV vision spectroscopy. Instead, the only difference is the incident light is circularly polarized. First, the left-hand the light is introduced to the sample and measure its observance just like typical UV vision. Next, do the same thing with right-handed light. Let's subtract observance difference between LCP and RCP. If there are non-zero signals like this in specific wavelengths, we can say that there is energy difference resulted from sample shear structure. Just like the typical UV vision, since there are observation peak data for well-known enantiomeric molecules, we can distinguish these molecules by CD spectroscopy. As shown in the bottom figure, by comparing obtained CD pattern, we can distinguish chemical species and their handiness. Moreover, by using CD spectroscopy, 3D structural data with specific handiness can be obtained for bioorganic molecules. The left figure shows the CD spectrum obtained from typical left-handed polypeptide molecules, and the right figure shows a spectrum obtained from DNA molecules. Likewise, UV vision can be enhanced and functionalized by substituting incident light source. And this is the end of presentation, and thank you for listening.